So, as you know, clinical trials are underway in this disease, and it's wonderful that therapies are finally emerging after, I've been talking to you for almost 2 decades now, and when I first started talking to you, there was nothing. So, but as you also are painfully aware of, they don't always succeed. And so they cannot succeed for a number of reasons. One reason is, as Jill's already alluded to and John's alluded to, sometimes the animal models themselves are imperfect models. So even though the drugs do exactly what we think they do, engaging the target they engage in the human patient may not have the same effect as it did in the animals, and so they fail for that reason. But all too often they don't fail for that reason. They fail for the reason of either the human trial was improperly designed because we didn't really understand the mechanism well enough, or in some cases, and what I'm supposed to really talk about in some detail today, is they fail because the preclinical studies were really not robust enough to inform us of their possible efficacy, the drug's possible efficacy, and how we really should design the trial. So translational research, as you know, really starts with the preclinical research, and that can be in the form of cells and animals, whatever models we've got to inform us of how to move on to do the clinical research, to give these therapeutics the best chance to succeed once we get into humans, and hopefully then move on to clinical implementation. Now, translation in DMD patients is particularly difficult because all that we know about the pathophysiology is really based on what we know in animal models. And most of the human data comes from the histopathology, which correlates to a large degree, but not totally, as I'll point out, with what we see in the animals. And so we're extrapolating quite a bit. So the big question is how do we extrapolate from these improvements that we see in mice to humans, and which parameters and which targets are most important that we see in mice, which ones are really going to be the most predictive of what we're going to see in humans, and how much of a change do we need to see in the mice before it's going to translate into something that's useful in a human. Now, Eric Hoffman, the speaker after me, is going to talk about patient biomarkers and the need to develop them so that we can actually make very early decisions in human trials as to whether we're engaging target and whether things are moving in the right direction, because that would also help, because we could either then drop a target or we could reevaluate how we're approaching the dosing or the testing, and then and try to save a drug that might be doomed if we carry out the trial the way we've designed it, but it actually could work if done properly. So, and then what John really wanted me to start out talking about is the fact that most of the animal studies that scientists do, especially in this field, are really more proof of principle studies than they really are true preclinical studies. So they actually are, most of them are not blinded, they're not randomized, and many of them rely on histological, non-quantitative parameters to evaluate what happened. Real preclinical studies need to be blinded, need to be randomized, and the parameters have to be quantitative. And if it's really going to foreshadow a human trial, then really the outcome measures need to be established ahead of time based on what has been seen in earlier studies. And even in that case, even if you do the best preclinical studies, not every lab's colony is the same, they're not treated the same, and so there are variabilities that make it sometimes hard to extrapolate from one set of experiments to another. So there are many difficulties in optimizing the predictive value of the preclinical research. Just to name a few is to reiterate this point that the mechanism and the target that we really have focused on in the animal studies may not really be all that disease-modifying in humans. So you might have done the best possible animal studies, but really the target you focused on might have been important in the mouse, but not so important in the human. You might just not have done the study properly. I mean, the PK or bioavailability may not translate, so you may have to readjust how you're doing the dosing. You need, in this case, and you need to really check early, you need to have biomarkers that you can look at and basically be able to sort of redirect the trial. And then one of the things I'm going to talk about is that I do think we do need better animal models. And so I think some of our drugs could possibly fail because our mouse models really don't model the human disease all that well. As John said, the mice don't have Duchenne muscular dystrophy. They're missing dystrophin, but we really do need better phenotypic models. And then in many cases, the studies really aren't done that well. The effect size is not robust enough. So it might have been a weak candidate in the first place and really should not have necessarily moved into humans. And then the last issue, which a considerable amount of press has been around the past few years, is the unintentional biases that investigators introduce into preclinical studies that doom the later human trials. They're not rigorous enough in their design and conduct, and really the sort of reporting of the data needs to be improved. And so NINDS in particular, John's former employer, has really taken this on as a major thing that needs to be fixed, and not only in our field, but in all of the fields where the preclinical work is meant to inform the future clinical studies. And this is a slide from John just showing that 29 different studies of a drug in preclinical stroke models showed very different results depending on what the parameters that were analyzed and reported were. And in this case, the more that they looked at, the less efficacy they saw. So this is a problem. So this really is not an issue of the right animal model. I mean, that's a different issue. But this is really the issue of if you've got the best possible animal model, you've got to design the best possible preclinical studies to look at any disease. And you have to avoid putting in unintentional bias into your analysis of the data. So the stakes are high. Obviously, if you're going to either move forward or drop targets that could be efficacious based on data that really didn't inform us, then we're just wasting time and money. And worse than that, we're putting patients into trials that they shouldn't be in, and we're just not making the best use of any of our resources, be it financial or human or the investigators. So what has really been sort of proposed for preclinical studies is more of a three-stage model, where we do start with pilot study, the real discovery phase, where you test the initial hypothesis and try to engage the target and see what sort of efficacy one sees, but basically to interpret that with some caution, and then move on to a more exploratory preclinical study where you really drill down on the mechanism and target in some detail. And at this point, it should be a truly randomized blinded study with a clinically relevant design to assess efficacy and multiple outcomes of efficacy, as well as to assess the target engagement. And then, and only then, move on to a study that truly mimics what the future human study might look like, a randomized blinded trial where now one has a predetermined outcome measure and multiple models if possible, and a firm statistical basis for analyzing the data. So in order to sort of help this, PPMD has invested in what we're doing in Florida to try to create an animal testing core with the goal of being able to evaluate the therapeutic potential of a number of interventions, novel interventions, approved drugs, whatever, and do it in a rigorous way to do these preclinical studies and preclinical trials in mouse models of Duchenne in order to help prioritize what therapeutic inventions really should move into the clinic. And this involves myself and Beth Barton, who directs the core for our Wellstone Center, and then a number of staff members, because one thing about doing these large blinded studies is you need a large number of people. And so this does, I think, for small labs make it difficult, because to really do these rigorous blinded trials, you need a large number of people involved in it. And so not that many centers, not that many labs really have the capabilities to perhaps do these final phase animal trials in the way they should be. And so operations like ours and Raju's at Children's National and a number of others do have those sorts of capabilities. And then the mouse models we use are multiple for Duchenne models, as well as multiple for other models, but in particular, we're using not only mouse models, but as well, we are also using the golden retriever dog model. So sort of the way we approach this is that we don't necessarily do the initial proof of principle study. So we do in some cases, but we usually are entering at the exploratory preclinical study phase where we're doing these blinded studies and testing something where there's already a hypothesis, already a potential target and hypothetical mechanism. We're trying to confirm that. And then if that looks good, we move on really in parallel to preclinical studies in mice, where we do more human-like study designs, larger numbers of animals, blinded, predetermined outcome measures. And in parallel, we try to then go back and do a confirmatory preclinical study in dogs, where we don't have the numbers of dogs to do larger studies, but we can do these studies in dogs where we confirm mechanism and target engagement. And again, those are done in a randomized, blinded fashion. Now, as I said, beyond the difficulty in actually performing the preclinical trials properly, in the Duchenne world, we also have the problem that the MDX mouse is not really a great model for the disease. And we've known this for a long time. And so one of the things we've done as part of this core that PPMD has done, and other people are doing too, is evaluating new animal models to try to see if we can find mouse models that are better phenotypic models. And just to give you a little bit of an idea of what that looks like, here I show the same muscles at the same age from 3 different mouse models, all dystrophin deficient. And on top is this sort of garden variety MDX mouse, and on the bottom is the DBA mouse, and in between is the normal C57 MDX mouse on the CMOD deficient background. And what you can see is as you change the model slightly, in one case by just one gene being knocked out, in the bottom case by a number of genetic variants, you can make the disease much worse. And we would say that we think the DBA mouse that background gives you a much more human-like disease in that you have a sustained inflammatory response that's much more severe than in the MDX mouse and is sustained in many of the tissues for a much more prolonged time, leading to much more fibrosis, and also a hallmark of the human disease histologically that we've never seen in the normal MDX mouse until they're very old, is we see a very early infiltration of fat into the tissue as well as fibrosis. So this really looks to us much more like the human disease tissue. And so this is the mouse that we're now sort of moving toward as our gold standard. If we can't impact the disease progression in this mouse, then we're not so sure we should be moving the therapeutic into humans. At the same time, we're still relying on the golden retriever for further validation of the therapeutic, because again, it has a disease that's much more human-like. This is just showing histology of several muscles at 18 months of age in the golden retriever, showing a normal dog's tissues on the left, and the untreated golden retriever in the middle, dystrophin deficient, and then tadalafil, which is in clinical trials and will finish at the end of this calendar year, just showing that part of the preclinical data package that we had that made us think that tadalafil should go into younger patients was this sort of prophylactic effect. These animals were treated starting at a fairly young age and sustained for a year. And what you can see is the disease progression in the skeletal muscles is much slowed. And we're hoping that a year trial in humans is going to show something very similar. So the other difficulties really, and Eric will talk about this a little bit, as I said, if the animal studies don't really give you the correct targets, then you're going to have to really do further mechanistic studies. But also, you want tools that can help you make very early decisions as to whether you should keep moving or whether you should change your study or whether you should just drop the therapeutic altogether, the so-called phase 0 or perhaps small phase 1 studies where you're really looking at target engagement and readout being biomarkers more so than even functional measures because of the short time period of these trials. And this has been another area that PPMD has really helped us invest in. And so in Florida, for over 10 years now, we've worked on this project along the lines of looking at the natural history of Duchenne by following the patients not only with measuring functional outcomes, but also with MRI in order to try to develop MRI as a biomarker. But in some ways, I consider it more than a biomarker because it's really looking at the status of the muscle, the health of the muscle. So you can follow the progression of the disease by MRI without the help of biopsies. And MR has a number of signatures that allow you to see various aspects of the disease. And of course, over time, you can see the replacement of the muscles with fat and fibrosis very, very easily with MR. And so we think the combination of MR with other biomarkers as well as appropriate functional outcome measures is going to allow us to have short-term early trials, small numbers of patients because the MR is highly reliable, not subject to any volitional input from the patient, and allow us to either triage a drug that's not working or redesign a trial to allow the drug to work. So that's what we're doing to try to optimize the chances for clinical trial success with the support of the Parent Project, with support of our Muscular Dystrophy Center from our Wellstone Center, and with support now that I have to establish a myology center at the University of Florida. We have this animal core, which is going to evaluate in a rigorous way the potential therapeutics. We're developing these non-invasive MR tools. We're looking at monotherapies as they're advancing, but also we're beginning, as Jill mentioned, to combine them in these animal models to try to figure out what are the most effective combination therapies. So as the monotherapies are available, we'll know which ones, or at least we'll take our best guess at which ones we should try to combine in studies in the boys. And then lastly, we're conducting our own early phase human trials to evaluate potential therapeutics, either approved or investigational ones, using MR as a major outcome for these early studies, along with some functional outcomes. So this is my acknowledgement slides, but as I said, this work is mostly, all of the work was started by Parent Project that I talked about that we're doing, and a lot of support from NIAMS and NINDS. And obviously a lot of what I talked about is what the community is doing, because the community's been doing a lot over the last 10 years. And if there have been a few setbacks, I think we've learned from them, and hopefully we will improve the preclinical studies, improve the clinical designs, and we won't make as many missteps. We'll try to bring the best possible drugs to the boys and get them approved as fast as we can. Thank you.